Hello, ladies and gentlemen, and welcome to today's broadcast on robust, reproducible protein quantification in plasma using Evosep One and Agilent 6495 LCTQ by Linfeng Wu. I am Abby Pell of LabRoots, and I'll be moderating today's event. Before we begin, I would like to encourage you to engage with us. You can submit as many questions as you want at any time during the presentation. Simply type the question into the Ask a Question box on the left side of your screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right or use the Ask a Question box on the left. It is my honor to please welcome Linfeng Wu.
Thank you, everyone. My name is Linfeng Wu. Today, I'm going to present our work for peptide quant in plasma using a new low-flow LC system called Evosep One, coupled to Agilent 6495 triple quadrupole LCMS with Agilent nanospray source. First, I would like to talk about targeted protein quantification in translational research. Nowadays, a trend in translational research is to study multi-omics, such as genomics, epigenomics, transcriptome, proteome, metabolome, lipidome, and their relationship with environment, and then study their interaction with each other. So the expression of these multi-omics result in phenotype. Today, my presentation is focused on proteomics. In biomarker evaluation and precision medicine, users often need to target a limited number of protein biomarkers and then monitor the expression change during a period of time. So this type of study often ends up with several hundred to even thousands of biological samples. This will require analytical technology which can be very reproducible and very robust. Multiplexing liquid chromatography mass spectrometry is a very powerful technology. It has been widely used for targeted protein quantification. To achieve best robustness and reproducibility, standard flow LC triple quad is a platform which preferred compared to nanoflow and microflow LCMS system. Here on the left is a picture of Agilent 6495 LC triple quad, coupled with a 1290 Infinity 2 LC and a Jetstream source. So this is a standard flow LC triple quad. It has a superior analytical sensitivity and very robust and reproducible. So it has been well accepted by the market for targeted peptide quantification. Here on the right is an example of MRI chromatogram from 99 targeted peptide using this platform. In this MRI chromatogram, the peptide retention time has a varied very differently, and the MRM chromatogram varied in a wide dynamic range. In terms of peak width, a median full width at half maximum is only 3.5 seconds from these 99 peptides. So you can see this LC platform can generate excellent peak separation. So using this platform, Agilent has generated a bunch of application notes, so it can be downloaded from Agilent website. Using standard flow LCMS system, users often need to load more sample on the column to achieve optimal analytical sensitivity, but sometimes users are sample limited. So in this scenario, a low flow LCMS system is often preferred. This is why we evaluate a new low microflow LC system Evostep One and coupled to Agilent 6495 LC triple core to supplement this scenario for people who are sample limited. So in this LC setup, we have Evostep One LC coupled to Agilent nanospray source and Agilent 6495 LC triple core. And all the data showed here in this presentation has been using this LCMS setup. Here on the bottom shows the screen capture of the nanospray monitored by the camera on this system. And in the middle is the picture of the clamshell, which it can hold the analytical column and the spray emitter. Here the analytical column and the spray emitter are provided by the Evostep company. And the spray emitter is a stainless steel emitter. And this resulted in very robust spray in our test. And this column and the emitter fit nicely into Agilent clamshell, which are going to the Agilent nanospray source. Evosep One is a standardized platform which was mainly designed for robustness and high throughput study. They achieved this by using a disposable trap column. Sample was first loaded and desorted offline on a pipette tip called the Evotep. Then the auto sampler will pick up the Evotep, place it in the injection port. Evosep One has 4 low pressure pump and one high pressure pump. They're using a gradient offset focusing technology to deliver the LC separation. So first, the 2 low pressure pump will deliver a gradient with a relatively steep slope gradient, which shown as a red line in this gradient plot. And the sample, after the sample eluted from the Evotep, the other 2 pressure pump will deliver another gradient which is relatively gentle gradient, and it will offset the first gradient and make a preformed gradient in the storage loop. Then the high pressure pump will push this preformed gradient in the storage loop onto the analytical column. So this integrating the illusion can reduce the injection cycle overhead, and they will remain the contaminant on the Evotep cartridge, which will be disposed after each injection. Since all the elution and gradient formation happened at low pressure, so they can minimize wear and tear. Evosep provides 6 methods which cover a range of user cases, from high throughput studies such as 300 samples per day to proteomics discovery type study, which will give 15 injection per day. So the LC flow rate can range from 4 microliter per minute to 220 nanoliter per minute. Using this system, we design experiment to evaluate their performance. Here is our experiment flow to evaluate this platform. We first took human plasma and diluted with ammonium bicarbonate. After dilution, we denature, reduce, and alkylate the sample, then trypsinize the sample. After trypsinization, we lyophilized the samples, and then we took a stable isotope labeled standard peptide mixture from a biomarker assessment kit, which was purchased from MRM proteomics. We rehydrate this SIS peptide mixture, spike it into plasma digest. We prepare 2 sets of sample for evaluation. The first set sample is is for standard curve analysis. We spike the sample at 8 different concentrations. We replicate. And the second set of sample is a large stock of plasma digest spiked with SRS peptide at 10 femtomole per microgram, and we with many replicates. So for robustness test, we continually injected replicate samples and to test the signal stability. We performed 3 experiments in this performance. First, we load all the sample onto Evotep before robustness test, and we inject the standard curve samples with replicates, which ended with 8 concentration. Each concentration have 5 replicates. And then on the same column, we perform the robustness test using the same sample. And after robustness test, we inject the remaining standard curve samples to create identical the standard curve, which similar as the experiment 1. Before I show the result, I would like to highlight a few things about the LCMS method used in this work. So in these 3 experiments, we monitored 33 pairs of heavy and light peptides, which totally gave us which totally has 198 MRM transition. So we use an analytical column, which provided by Evosep. The column is 8 centimeter long and 100 microgram ID. And the flow rate we use 1 microliter per minute. The LC gradient length is 21 minutes, including the injection and the post-gradient washing. The cycle time is 24 minutes. So this will allow us to inject 60 samples per day. Including sample method development and performing all the 3 experiments, we continually operate the system almost 12 days. This gave us a total 654 6 sample injections. For each injection, we load about 1 microgram plasma digest on column. So during this test, we didn't change the column. All the experiments were performed on the same column. And we didn't change the spray emitter and didn't adjust the spray voltage. So, and also we didn't tune or clean the mass spec system. For convenience, I would like to show the robustness test results. In this test, we performed totally 574 replicates and monitor 33 pairs of peptides. So here shows the retention time distribution for all targeted peptides. The different peptides were coded in different colors. So from this plot, you can see the retention time quite stable throughout this test. That means the analytical column back pressure was not increased during this stress test. And there's only a little bit bump in the middle. So the retention time CV for all these peptides is ranged from 0.4% to 2.7%, and most peptide has a CV below 2%. Next, we'll look at the peak area. From the relative standard deviation histogram, you can see a median of ISD, a median ISD 8.5% was achieved, which was represented by the blue dashed line here. And 94% of peptide has an ISD below 16%, which was represented by a red dashed line here. There's only 2 pairs of peptide has relatively high ISD, and we examine these 2 pairs of peptide. One pair of peptides are degraded fast. They are not very stable, because we observe more than 50% degradation after overnight incubation at 4 degrees. The other pairs of peptides, we observe very severe background interference. So we have to manually integrate the peptide, which causes a lot of variation. So from this result, we conclude that these two outliers are not due to platform variation. And here shows the MRM peak area distribution from 4 selected peptides. And these 4 peptides are represented by different color. And each dot means one replicate injection. And overall, you can see the MRM peak area is quite stable. And these 4 peptides, the highest response peptide has about 11-fold signal response compared to the lowest response peptide. And these stable isoto-labeled peptides are mixed at an equal amount. So you can see from the bottom 3 peptides, their signal response has about 1-fold difference from each other. And from these 4 peptides, you can see their peak area IST range from 6% to 7.9%, and their retention time IST range from 0.59% to 1.04%. And they are quite stable if you consider this is running at 1 microliter per minute. And next, I would like to show the standard curve analysis, which performed right before and after the robustness test. And for both standard curve, we prepare the sample at the same time, and they all have A concentrations, and each standard curve we inject 5 replicates for each concentration. So we try to generate identical the standard curve before and after robustness test. Here shows the 2 standard curves from the same peptide which match to complement C3. From these 2 standard curves, you can see they look very similar, and both have R squared close to 0.999%. And the standard curve concentration ranging from 4 atmol to 100 femtomole on column in plasma matrix. When you zoom into the bottom low concentration area, you can see both standard curve achieve a lower limit of quantification at 10 atmol on column. So here we define lower limit of quantification by using a criteria of IST lower than 15% and the accuracy between 80 to 120%. We next look closely on the data quality from these 2 standard curves. Here on the left shows the MRM chromatogram from the low levels using these 2 standard curves. So here shows the MRM chromatogram from matrix blank, 4 atmol on column, 10 atmol on column, and 20 atmol on column. You can see even at these low levels, this platform can clear detect chromatogram peaks. On the right is the table calculating the precision and the accuracy using these 2 standard curves. You can see at the lower limit of quantification level, which is 10 atmol at column, this platform can achieve about 10% IST, and the quantification accuracy is between 20 to 120%. At all the tested levels, we can achieve a pretty good precision and accuracy. All the data showed in this presentation has been published as an application note. If you are interested on the details, you can download this application note from Agilent website. With all this data, we concluded that using this Evosep One coupled to Agilent 6495 LC triple core, we can achieve very reproducible LC retention time for a high throughput study. And the MRI signal response showed very stable signal throughout almost a 12 days operation without any adjustment on LCMS system. And we can achieve a similar standard curve for the same peptides before and after robustness test on the same single column. Since we upload all the samples on the Evo tips before robustness test, and then we store the tip in a 4-degree refrigerator, we didn't see obvious sample degradation during this process, so we can conclude that the sample storage on the Evo tips are quite stable. In the end, I would like to thank our colleagues at Agilent Technology and our collaborator from Evosep, especially Nikolai Budget. He helped us doing this testing experiment and also deliver a training at Agilent site. Thank you very much. With that, I'm ready to answer your question.
Thank you, Linfeng, for the very informative presentation. And at this time, we will continue with our Q&A portion of the presentation. Just as a reminder, you can submit a question to the Ask a Question box. All right, we have a few questions that are coming in. And I want to first ask you, Linfeng, the first question is, are the LC software and MS acquisition software integrated together? And since
go ahead.
And the second portion of that question is, since the LC and MS instruments are from two companies, how many computers do you need to control the LCMS MC platform?
Okay, so the LC and MS acquisition software are not integrated together. They are standalone software, but you only need one computer. So these two software installed in the same computer. And then the sample work list was entered in the software called Kronos. And from this software, you can enter your sample information and select the LC and MS method respectively. And the two instruments were connected through a remote cable.
Great, thank you so much, Linfeng. And our second question is, can users create their own LC methods using Evosep?
Currently, the answer is no. So right now, Evosep provides 6 LC methods, which cover the different user scenarios from high throughput study, for example, like 300 sample per day, to discovery type study to with 15 samples per day. So the flow rate range from 220 nanoliter per minute to 4 microliter per minute. But the user can now customize the method right now by themselves.
Great, thank you so much. And I think we have a little bit more time here for a couple more questions. Our third question is, after loading samples on Evo tips, will the tips dry out?
This is a good question. So after sample loading, I add some solvent A in the tip box so that the top of the tip was soaked in solvent A. And this can prevent Evo tips from drying out during your your long-term storage, and also during the data acquisition.
Great. And okay, and I believe John here has a comment.
Yeah, I'd like to just offer a quick comment if I could, and that is that Agilent has an agreement now with Evosep. It's a co-marketing arrangement with Evosep. And just 2 quick comments on what drove this. We really see Evosep as helping to solve some of the very sample-limited applications with robustness, ruggedness. The issue in nano and 0.1 millimeter columns, 2 big issues that they've solved. One is when you get let's say a 96 well plate and you had 1 bad sample, it oftentimes would then cause contamination throughout the entire system, such that if you had 1 bad sample on most conventional nano systems, you'd be down cleaning, flushing for days and days. With Evosep having a cartridge for each sample, if you get one bad sample, your primarily contamination is oriented in that one sample. So that's one nice problem they solve. They also really help with transfer of methods. Their ability to transfer methods is greatly enhanced over other traditional nano systems. So we'll have a lot more to come on this as we go forward, but we're very excited with the relationship with Evosep and the co-marketing agreement. And Linfeng's been a big part of helping us get this all going.
Thank you so much, John, for that wonderful information. If we have no more questions today, we will be just ending the presentation and wrapping up a little bit early today. But as a reminder to our audience, questions we did not have time for today, and those submitted during the on-demand period will be addressed via the email address you provided at the time of registration. And just want to thank you again to Linfeng Wu for your presentation and your research. And to everyone who participated in this presentation, we hope you enjoy the rest of today's event.